And I will introduce myself as the next speaker. So I'm Adi Stern from the Faculty of Life Science. And so my talk will be about, I have to track time. Yael, will you help me track time for myself? What makes a variant and drivers of evolution during chronic SARS-Coroanvirus-2 infections. And so we really don't need the introduction here about this virus and about its genome and its similarity to other coronaviruses. And of course, the importance of the spike protein in mediating the interaction with the receptor and being the target of the antibody response. I will talk about the evolution of this virus. And so pretty early on in the pandemic, the rate of evolution of the virus was calculated as being 2 mutations per month, which means that a mutation is fixed somewhere in the genome of the virus every second week. Okay, and so just to look at this toy illustration, we have a person infecting another person, and we don't see any mutations. And then the second time there's an infection, we see a mutation. And this goes on, and so mutations accumulate, okay, at a relatively fixed pace. And it was quite amazing, actually, to look at graphs showing how the virus evolves a long time during the first more or less year of the pandemic, and seeing that really there's this clock-like evolution. Mutations accumulated more and more and more. And we could actually, if you'd have given me a sequence of the virus at any time point, I would have more or less been able to tell you how long, when this sequence actually infected someone. And let's go on to talk about variants. Okay, so technically, a variant is actually any genetic variation. So any of these mutants that we see here, okay, in this illustration, is actually a variant. Okay, so that's technically speaking. But of course, in the way we the media or the public speaks about a variant, we actually mean a variant that has some dramatic change in phenotype. And the World Health Organization, as well as other health organizations, define 3 categories of variants. So there are variants of concern, variants of interest, and variants under monitoring. Currently, there are 5 variants that are considered variants of concern based on the fact that they have either increased transmissibility or increased virulence, and or a decrease in vaccine or drug effectiveness. And these are alpha, beta, gamma, delta, and Omicron. There are other variants of interest that actually also showed some of these, there was some evidence for some of these phenotypes, but they were variants that actually usually kind of emerged and disappeared very quickly from the world. Okay, so all variants of concern, one of the first characterizations when looking at their sequences is that they had many mutations, many more than were evident at that time point. And so, for example, when alpha came out, at the time alpha came out, if you would have looked at a sequence that was not an alpha sequence, we would have typically seen something like between 5 and 10 mutations on the genome at that time, or a little bit more, but more or less. And you can see that alpha has more mutations, okay? So it had more mutations than expected. And all of the variants had more mutations than expected based on the molecular clock that I showed you before of 2 mutations per month. And not only that, the distribution of mutations was different. So most of the mutations were in the spike proteins, and most of the mutations were non-synonymous mutations, so mutations that change the amino acid as opposed to synonymous mutations that don't change the amino acid. And so this was already the enigma that came out. Where are these variants coming from? And this is what Omicron looks like. Okay, so you can clearly see that it's off the charts. And I'll just share with you, a kind of small story. Is that when Omicron was first discovered, it came out on Twitter, social media playing a really dramatic role here in this pandemic. And I was talking to my collaborator, and I said to him, "I'm sure this is a sequencing error. There's no way that this variant could be for real." And of course, it turned out that I was wildly wrong, and it was very much for real. And you can see the large number of mutations, especially, again, the huge number of mutations, so 30 mutations in the spike protein, as Natalia also mentioned. And so this leads to the question, okay, so what makes a variant? How is a variant created at all? Because 2 mutations per month is really way too slow for a variant of concern to be formed. And sort of hints at where this was coming from started coming out at the end of 2020. And these were these very rare immunocompromised patients who developed chronic infections. Okay, and so typically an infection will resolve within 2-3 weeks, sometimes a month, but some of these patients were infected for months. And we in fact, recently a patient came out that was infected for over a year. And this was, this was really, really worrying and surprising. And first of all, clinically, these patients are at much higher risk, naturally, because the infection does not resolve, and some of them really deteriorate due to the virus itself, not only due to the background condition, and some of them exhibit very, very rapid evolution and antibody escape, and soon we'll discuss this. And so when the alpha variant came out, the first hypothesis is that maybe really this variant came from an immunocompromised individual who was treated by plasma or monoclonal antibodies, and this led to antibody escape. And so there was a strong selection pressure in such a patient. And again, so there's the lack of the immune system on the one hand. On the other hand, these patients are treated with a fixed or more or less fixed antibody treatment. And this leads the virus, this selects for variants that escape either the plasma or the monoclonal antibodies. And so this was kind of the first hypothesis on how variants are created. And I'd like to share with you two enigmas that we studied in this context. And so the first enigma is that patients are actually very, very different. These chronically infected patients are very different. Some patients, we see very dramatic evolution. Some patients, we actually see almost no evolution at all. So the virus is almost static for months. The second enigma is that we began this as a collaboration with Ichilov and with Dr. Eyal Paran and Susie Myers, and they told us about these cases that they have where there's this cycling viral load. So the virus seems to clear, and the patients are released home, and everyone is happy, and the virus, the COVID is resolved, and suddenly 2 weeks later the virus returns, and this happens over and over again. And we really couldn't understand what exactly is going on in these patients, and this was clearly of clinical concern. Because when these, if the virus is still there, if these patients are being released and the virus is still there, this is of course very risky, also for their own sake and also for the sake of the surroundings. And so our first question was, what predicts rapid evolution and immune escape in patients? And what we did here, and this is a study led by Sherry Harari, it's already out on medRxiv, and it's in revision, the paper. And so what we did is we gathered all the case reports. So until at the stage that we came into the study, there were these isolated case reports. Everyone that found a case, found one of these cases, published it as an individual case reports, and we put them all together. And we also have 6 patients here that we sequenced, again, from Ichilov, and they're part of this cohort. And so this is a background condition, and you can see that all of them have some kind of immunocompromised background. A lot of them are these hematologic cancer patients, but not only. There are other patients here. There are AIDS patients. There are autoimmune disease patients here, and so there's sort of a variety of backgrounds here. We looked at their their background treatment. And so a lot of them were treated by CD20 antibodies, okay, which basically remove, B cells from their bodies. And this is actually a very, very efficient treatment, but clearly it creates a problem, okay? And this is most likely what is leading to the fact that these patients cannot clear the virus, okay? But you will note that this is not true for all of them. Some of them were not treated with this background treatment. But all of them did have some form of immunocompromised background. Some of them were treated with steroids. Some of them were not. Some of them were treated with antibody-based COVID treatment, what I mentioned before, either convalescent plasma or monoclonal antibodies. And so you see the pattern here is very, very mixed. And this is the outcome from our point of view. And the outcome is the ones that are marked here in green are those where we see very, very well-known mutations that are associated with antibody evasion. And so what we did here first is we performed a logistic regression to try and see what feature actually predicts or eventually predicts an outcome of an antibody evasion mutation. And so actually none of these features here, which is either anti-B cell background treatment, steroid treatment, or antibody-based COVID treatment, none of them predicted antibody evasion. The only thing that did predict, in a sense, predicted antibody evasion mutations was viral rebound. Okay, so the situation where the virus disappears and then reappears, and disappears and then reappears. So in these situations, and I'll show them here, what we see is that when the virus returns, it returns with an antibody evasion mutation. Okay, and so you can see this here. The x-axis is time, the y-axis is viral load. And so you see, this is a patient that drops, right? The viral load drops, so the virus is cleared. When we're in the red zone here, that means that the virus is cleared. And then it goes back up again, and when it goes back up again, it is accompanied with an antibody evasion mutation. And so this really means that we have to be very, very cautious with these patients, because it means that when we're releasing them, the body might not have cleared the virus, and when they return, they're returning with something which is already the beginning of a new variant. Okay, so antibody evasion is one of the features that is really common to almost all the variants of concern that we know of today. The next thing that we saw is that all these patients were characterized by highly polymorphic populations. So we see mutations come and they go and they disappear and they go up and they go down. And this was very actually surprising evolutionary-wise, because usually when you see a mutation that is present, again, at a very high frequency in the population of viruses that we're sequencing, so it's fixed, it's there, how does it disappear? Where does it disappear to? Does this mean that another, that it reverted, that a mutation sort of reverted backwards? And that, statistically is very, very improbable. And so what we think is going on here is that the virus is migrating between different regions of the body. And so actually in one of these patients, we know that while the patient was negative from a nasopharynx swab, so that's a typical swab, when isolate from lungs was extracted, the sample was positive. Okay, and so this suggests to us that the virus, the fact that it's disappearing, it just means that it's disappearing from a nasopharynx swab, but actually the virus is continuing to replicate in lungs, and then it might be migrating back and forth, and so we have these very dynamical populations. And we also think that antibody escape mutations might occur in niches in lungs where there's very, very poor penetration of antibodies. And the reason that we're suggesting this model is that similar dynamics actually occur in HIV in the context of drug resistance, where you have a niche in the body of a patient where the drug doesn't penetrate properly, and then you get resistance. The second part of the talk is going to be what makes a variant beyond antibody evasion. And so we know antibody evasion is not everything that makes a variant of concern. And so first of all, what we did is we compared here chronic infections. So again, the x-axis here is the genome position. The y-axis is counts. And so these are, the upper panel here is all our chronic infections. And we just counted, where did we see mutations? Where were mutations occurring in the chronic infections? And the pink region here, the pink background is the spike protein. And so very, very not surprisingly, we see a lot of mutations in the spike protein. And we compared this to variants of concern. And you can see the similarity, okay, in the profile overall. So the same regions in the spike protein that are highly mutated in chronic infections are the ones that are also present in variants of concern. And so chronic patients as a set are somewhat a predictor of the mutations that we're going to see in variants of concern. But what was interesting actually was the differences. So some mutations seem to be missing. So for example, this mutation that is present is, in variants of concern, it's an ORF1 of the virus, was absent in our chronic set. Not only that, mutations here that are at the S1, S2 boundary of the spike protein, a region that is known to be very, very important for the virus, and a region that we see is highly mutated in variants of concern, we didn't see any mutations in our chronic set. And then this study came out just recently, and this study actually measured the effect of mutations on transmissibility of the virus. And I won't get into how they do this, but what they noted is that, what we noted is that exactly the set of mutations that they found to be most transmissible, so mutations that are associated with very high transmissibility, are the mutations that are missing in our chronically infected patients. And so what we think this means is that actually when a virus is replicating inside a chronically infected patient, there doesn't have to be selection for transmission. The virus is happy inside this chronic infection. It's got an endless reservoir of cells to infect and go on, and nothing is in its way. And so there's no selection for transmissibility. And so I think this makes sense. And this led us to come up with a model of how does a variant of concern eventually come up in a chronically infected individual. And so there could be the lottery model, which means, okay, so we don't have selection for transmissibility, but maybe 1 in a million, and I have no idea what the number is, 1 in a million chronic infections, a transmissibility mutation just comes up, and this leads to a variant that is very transmissible that moves on. The other model that we have is that there's maybe a conflict. That maybe there's a conflict between mutations that promote transmissibility and mutations that promote antibody evasion. And so maybe that is slowing down the generation of transmissible mutants in these chronic infections. And if that is the case, that is, of course, better news for us as humanity. And so we see some preliminary evidence for this model because, for example, E484K, which is the most well-studied mutations in this spike mutation in the context of antibody evasion, we very rarely see it ever together with P681H, which is a mutation that is present, P681H, or R, a mutation that is present on almost all variants of concern. And so this is still work in progress that we're trying to follow up on. And with that, I'll summarize. I will say that regardless of the mechanism, the more infections there are, right, and so wave 5, the number of infections worldwide was simply huge, the higher the probability that we're going to get an additional variant of concern from some chronic infection. Okay, so I mean, I just, I keep thinking about the number of people that were just infected now, and of that individual that might now be developing the new variant, which will be responsible for either, I don't know if we are already in wave 6 or wave 7 or whatever. And clearly we need to understand the evolution of SARS-Coroanvirus-2 and chronic infections better. And so I'd like to thank the wonderful people who did the research. Sherry Harari led this front across extensive bioinformatic work that we did here and collaboration with Tel Aviv Sourasky Medical Center, yes, Yael and Susie, who I already mentioned, Ronen Amos, and all the rest of our collaborators. Mayan and Natalie were also instrumental in this work, and the funding agencies, of course, and thank you for your attention. I'm happy to take questions. Yes, that's true. So yeah, I'll repeat the question is about T cell treatment. And yes, some of these, we tried looking at that as well. Some of these have CAR T cell treatment. It was not a predictor either, but it is possible that, maybe that there are complex, we didn't always have the full medical background of the patients. And so again, this is working with limited evidence. And we didn't see it as a predictor in any case. Yeah, that's an excellent question. So I know from previous studies that yes, that the virus is often observed in the blood. Some of these samples were actually from very few samples here were from the blood. We never were able to, so a very, very interesting question would be to compare between different organs and see if the profile of mutations is different. We don't have enough of a sample size to do it at the present, but I think that would definitely be something to look into in the future. Yes, I agree.